This video is going to be the last two sections of chapter 6. It's isolation, culture, and identification of viruses. And then we're going to go, the last section will be the more acellular stuff, but not viruses. It'll be the virusoids, viroids, and prions. So let's get started on this. The first one is talking about isolations. So remember, viruses are super, super small. We talked about that in the very beginning. We saw the epithelial cells of animal cell, and then a bacterial cell, E. coli, and then the virus on the side of the E. coli. So they're really, really small. How do we isolate those? We mentioned it in the very beginning too when we talked about tobacco mosaic virus, and Pasteur's porcelain filter would keep the bacteria out, but it wouldn't stop the viruses from getting through. So the filter would only allow teeny tiny little things through it. So instead of porcelain filters, now we can use membrane filters that'll do the same thing for us. So on the membrane filter, we can take a solution of the cells infected with the virus particles, and we'll take that solution of cells infected with the virus particles, we'll spin it down a centrifuge, and the big stuff like the cells are going to go in the bottom, and the lighter stuff like the virus particles will be on the top. So we'll take that top solution and we can put it through a membrane filter. So here the pores are 0.5 micrometers, and we can see that we've blocked out the red colored substance, but the blue and the green colored substance are still getting through at 5 micrometers. And then we decrease the size of the pore to 0.2 micrometers, and now only the little bitty green stuff gets through. So that's how we can use this. We can use membrane filters to get down to just the teeny tiny little viruses and isolate those. How do we culture them? To culture them, we can use cultures of cells in vitro outside of the body, or we can use a whole organism and all of its cells inside of it. So in vivo or in vitro. Uh, when we want to use it and culture it in a plate, we got to do a little of this in lab, but not all the way. So you take your Petri plate. This is a PEA and not just our normal growth medium. So this is phenylethylamine alcohol agarose instead of just our normal agarose that we're using that grows everything. This is a special one and selective and differential media, but I put it in here so you could see that it was clear and see-through all the way through. And you got to play with these in lab and see that they were clear and see-through. What we didn't get to do in lab is lawn an entire plate. So when you lawn an entire plate, you're going to coat it with that bacteria, and that bacteria is going to grow. So in 2 or 3 days, you should see that bacteria grow and take over the plate. And it's no longer clear, it's cloudy, like these guys. So the bacteria has grown on the agarose, it has made it cloudy, and it's no longer see-through. So what do? If this bacteria then gets infected with a virus, we can watch this virus start to attack the bacterial cells. So what we do is we take our agar concentration and we dilute it from 1.5%, and we're going to drop it down to 0.7% agar. So we're going to water it down. When we water it down, it makes it thinner and it makes it easier for the virus to get through and infect all these different bacterial cells. And when it does, remember we talked about the 2 life cycles in the previous section. We talked about the lysogenic, where it lurks and it's a latent infection, or the lytic cycle, where it just destroys. It takes over, it conquers, it destroys, and it lyses the cells. So that's what we're looking for here. We're looking for a lytic cell infection, and when it ruptures the bacterial cells, it makes a little clear spot next to the bacteria. So we can see these things. They're called plaques. And you can see these plaques all in this Petri dish. They're everywhere. So we've got plaques, a lot of plaques in here, and we have less plaques in here. So it's being diluted as we go. And then we've got a couple over here. So we can see these and look for this plaque formation where those cells have been killed off on these plates. So this is a way for us to culture it and then watch it in action, destroying these bacteria on the plates. If we were going to study this in vivo, why would we want to even look at this and study this? Why would we look at the viruses? Well, we want to be able to identify exactly what's going on with this. Is it this virus or is it this virus? So if we're trying to figure out what's going on and we want to figure out exactly which virus is attacking it, we need to study it. And so we would want to use this for identification if we are trying to produce vaccines or just research in general on that specific vaccine. We got to do studies. And so in vivo studies are where we're going to use a whole organism. So in this example, we have chicken eggs, and we discussed earlier that viruses are specific. They would be host specific, and sometimes within the host, they're tissue specific. We saw the picture of the human body and all the tissues all over the place that the viruses really like to attack. It's the same thing with this chicken egg. If we're going to infect the chicken egg, maybe we'll go for the amniotic membrane, maybe we'll go for the fluid inside the membrane, maybe we'll go for the yolk sac and inject that. And then we're going to see if that happens to do anything to the development of that chicken. Is it lysing the cells in there? Is it messing with the membrane? Is it messing with the growth and development of the chicken? What's going on? And we can study it. So in vivo are going to be studies of a living organism infected with the virus. In vitro are going to be the cells themselves outside of the body of the organism. So we would take those cells, we would culture them, scrape them off, whatever we need to do to get a hold of a sample of the cells, and we can do a culture. So we got our lung tissue, we're going to do a primary cell culture, and primary is the first one. So you take those tissue, the cell, and you're going to spread those cells out onto your petri dish, and those cells are going to need to attach. They're going to need a bottom and a top. So they are going to have what's called anchorage dependent requirements, that they're going to have a bottom surface. So you spread them all out on the bottom of a petri dish or a glass beaker, some flat surface that they can attach to, and then they're going to undergo mitosis, and they'll grow and they'll grow and they'll grow. But eventually, on normal healthy cells, you're going to have something called contact inhibition, where they realize there's more cells here than we need to, and we're packed. We don't need to grow any more cells, and they'll stop and shut down mitosis. So these are normal cell cultures. You got a primary cell culture, it's going to spread out, it's going to grow, but eventually contact inhibition happens. So you got to go and make a secondary cell culture to still try and understand and figure out what's going on with those cells. But if you use a cell where something is going wrong with this, there's a mutation in that cell, it's called a continuous line cell, and these are typically cancerous cells, cancer tumor cells. Those things don't have those restrictions. Cancer tumor cells aren't going to be restricted by anchorage dependence. It's not going to have a flat surface. They're not going to care how many of their buddies are around them, because the whole point of cancer is uncontrolled cell growth. And so you can see that there is a ton of cells on this petri dish. They're growing on top of each other. They're all over the place, and they will continue to grow. That's a continuous line cell. So we can try and use continuous line cells to study things, because they're not restricted like our normal cells would be. So I'm really glad the textbook here discusses this, because some of our other biology textbooks don't. But one of the first continuous line cells out there ever were HeLa cells. And those HeLa cells come from a woman named Henrietta Lacks. So we got He and La are the HeLa for Henrietta Lacks. So Henrietta Lacks was a lady back in the 1950s. She was married, and she was not feeling good. She knew something was wrong, something was bad wrong, but she didn't know what it was. And so she went to the doctor, and they took cultures, and it turns out that she had HPV that her husband had passed on to her, and it gave her cervical cancer, but she didn't know what she had. And they took the cultures of her cells, and they didn't really tell her what they were doing at all, or what they were going to do with it. But this was in the 1950s, so they didn't have any laws like we have now. So they took her cells, and when they cultured her cells, they kept growing and growing and growing, like we talked about, like these guys. So this was a breakthrough back then. They didn't have any luck with cells doing this in the lab beforehand, and her cells kept growing. So it was a continuous cell line. And the scientists were all excited about it, so they passed it around to all of their other scientific friends. They grew some and sent it to their friends, and they grew some and sent it to their friends. So everyone was able to use these cells and make some amazing breakthroughs from Henrietta's cells. We got the polio vaccine was studied using HeLa cells, and a whole bunch of other things have been studied using HeLa cells. They're still around today. Literally, she's immortal. The Immortal Life of Henrietta Lacks is the book that talks about this. So I want everybody to read that book. And if you don't like to read the book, watch the movie. it is a great, great story talking about Henrietta, and it tells you about what was happening with her life and how she was treated. And it is mind blowing the way that she was treated. Not only did she get improper medical care, they took those cells, they didn't tell her that they took her cells, and they didn't tell her what they were doing with her cells. And all of these breakthroughs, all of these discoveries made using her cells, they didn't let her know that they had taken them. They didn't ask for permission, they just did it. And then these companies are making money with the vaccinations that they're creating off of her cells. Did Henrietta know about this? No. Did her family know about this? No. No, they didn't get any compensation for this, nothing. They got a really raw end of the deal. And especially so, she was treated even worse because she was a person of color. So The Immortal Life of Henrietta Lacks is an amazing book. I recommend everybody read it. And it tells you the true life story of what happened with Henrietta and that we are still using her cells today, that she is immortal. It's a good story, so check it out. Immortal Life of Henrietta Lacks. HeLa cells are a continuous line cell that will grow and grow and grow, and we can use these things to study. So super cool, although it has a very not cool backstory. On cells that are affected with viruses, sometimes you can look at the cell and see. And if you can look in the cell and see that there are effects, those are called cytopathic effects. So on some of these things, they are going to have things called inclusion bodies, and different cells will react with the virus, and they may create an inclusion body. They may create, this one's got inclusion bodies, this one does too. Pox virus, herpes are going to cause inclusion virus. Herpes also causes cytoplasmic stranding, that you can see how it's almost like an arrow, like it's getting left behind as the cell moves on, or it's shooting out almost like an amoeba. It's got this streaming going on of the cytoplasm. So that's an indicator. It's a cytopathic effect that that cell is infected with herpes virus. Adenovirus also has cell enlargement, so the cells aren't their normal size, they're too big. So these are cytopathic effects. You take a sample of the cells and you can see what's going on. Are there inclusion bodies? Is there an extra looking thing that looks like a nucleus that shouldn't be there? All of these are cytopathic effects. Another test that you can use to try and figure out what's going on. If you've had anatomy and physiology too, we talked about agglutination. We talked about how red blood cells have these antigens on the outside of their cells, and there are antibodies that are cued in specifically for those antigens. Your body is going to make antibodies against things to fight them off, and those antibodies are like a lock and a key. So the antibody is looking for an antigen, and they will link up together. So lock and a key kind of thing. With red blood cells, if you're testing somebody's blood type, you'll take the sample of their blood and then you put in a solution that's got antibodies to certain specific types. And if those antibodies link up with that red blood cell, then you know that that blood is that very specific red blood cell type, and it will cause something called agglutination, and those red blood cells clump it together. If you don't see agglutination, then you're not getting reaction, and that person's not that blood type for whatever those antibodies were, whether you're A, B, AB, or type O, where you didn't get any kind of reaction. For the hemagglutination test, it's that same principle. Viruses are going to have antigens on the outside of them that can stick to red blood cells and cause the red blood cells to clump up or agglutinate. So with the hemagglutination test, we have normal red blood cells, just plain old red blood cells. They're not going to, when we have the results over here in this category, normal red blood cells, and there is no reaction. I'm not real big on this image, by the way, out of this textbook, because it's saying that on the hemagglutination test that there is nothing in row A, they're not binding together. So when they don't bind together, they sink to the bottom. I just don't like the visual. I want it to show no agglutination means that they're spread out, but that's not how it's showing in the well. So if this is what the well really looks like with this test, we'll go with it. But it's counterintuitive in my mind. I want it to look like the one below. But if we don't have any agglutination, it looks like this with this test. If we do have agglutination, so now we're introducing viruses, and the viruses have those spikes, those antigens on the outside that are going to link up with these guys, the red blood cells, and it's going to cause them to clump. And now we've got hemagglutination, And hemagglutination is going to cause it to be like a lattice work, and you can see it's different color. It's not all in the bottom of the well. It's spread out in this lattice formation with the hemagglutination. If we were going to use the hemagglutination test, we can use those antibodies that I told you are specific for that antigen. So these antibodies, instead of testing for A blood type or B blood type, these antibodies are testing for a virus, a very specific virus. So if you put these antibodies into the mix with all these things, then your antibodies are going to hook up with the viruses. They clog the attachment points. They're going to block the attachment points on the viruses, and the viruses can't stick to the red blood cells. So they remain separate, and we don't have, it's inhibited, we don't have hemagglutination happening here. So this is the hemagglutination test. If we add in the antibodies specific to that virus and those things are present, so the antibody is waiting for the virus, the lock and the key come together and they stick together and the red blood cells don't stick together, you're not going to have hemagglutination and it'll stay in the bottom of the well. So this is the hemagglutination test. Without the antibodies, the viruses stick to the red blood cells. With the antibodies, the viruses are stuck to the antibodies and not the red blood cells, so you're not going to get the hemagglutination. Another one, the nucleic acid amplification test, the NAATs. These are things where we're looking for nucleic acids, RNA, DNA. And we said every virus is going to have some sort of genome, RNA and DNA. Well, hopefully we mentioned in previous biologies, but if you didn't find out about previous biologies, there's something called PCR, the polymerase chain reaction. And what that is it basically copies nucleic acids. It's like a copy machine where you can make copies and copies and copies of very specific sequences of DNA. And so we can use the PCR machine to look for very specific segments of viral DNA. And so we can run this test through and see, do we have this viral DNA present? And that's a form of test. Or we could do the reverse transcriptase, where if it's a virus, we said viruses could use DNA, or viruses may also have RNA. So if we got RNA, we can use reverse transcriptase PCR, RT-PCR, which is what we're using to test for coronavirus right now. And that can look and see, do we have RNA present in this virus? Does this person have that specific RNA that is keyed for coronavirus or whatever the virus is that we're looking for? Are we looking for RNA? We'd use RT-PCR. If we're looking for DNA, a virus that has DNA inside, we'd look for just a normal PCR kind of test. So those are two different things that we can use to test for the genome inside the virus. Another thing that we can look and test for is enzyme immunoassays. So we are going to have a surface here, and on the surface, you are going to have, this guy does not have on the membrane filter, this is no virus over here, and we have virus over here. So we're gonna have two different samples, one that's gonna be a negative control and one that is gonna hopefully test positive, and we'll see. We got the virus, it's gonna stick onto the membrane. If we didn't have a virus, it won't stick onto the membrane. Then the next part is antibodies. So the immunoassays are going to use antibodies. And so here is the antibody, and the purple part is the enzyme on top. So the antibody is attached to the enzyme. Then we're going to have a substrate reaction. That substrate reaction is going to cause a color change. So on the first step, we have virus on the membrane. We don't have virus over here for our negative control. The antibodies and the enzyme, the antibody enzyme attachment happens if you've got the virus. Over here, we didn't have any virus to attach to, so those antibodies have nothing to attach to on this side. They have attached and stuck on here, no attachment over here, antibodies get washed away. Now we're on to step 4, where we add in the substrate, and when we add in the substrate, color change happens if this guy is still here. If we didn't have this guy, there's nothing to change. There's no color to change because there's nothing for it to react with. So this would be a negative test over here when you have nothing going on. And this color change would show you a positive test. So it's very similar to the urine stick test for pregnancy. Do you have HCG hormone? If you do, that pregnancy test is going to show you a line. And if you're missing the HCG hormone, it's not going to bind on there. There's nothing for it to bind, and there's no color change happening, so you get a negative reaction. So we're going to talk more about enzyme immunoassays in future chapters, but this is just kind of a primer on how you can use those to see if you've got a virus. If you got the virus, the antibody and the enzyme attaches, and then you hopefully have a color change. If you don't have the virus, there's nothing for the antibody to attach onto, and you're not going to see anything. Case study update. Okay, so we were looking to see if rabies was present in David. They ran the RT-PCR that we were just talking about. So rabies is RNA, not DNA. So we need to do an RT, reverse transcriptase PCR test. So we collected the saliva. They collected the hair and the skin sample. And his test is negative from the saliva. So we will talk about testing in the future, and we'll talk about negatives and false positives, stuff like that. But he doesn't have enough, pretty much. He doesn't have enough viral particles in the saliva to test positive yet, which is a good thing because we said once he starts showing signs and symptoms of this, rabies is fatal. So right now, his viral load is low, and his saliva test was low, despite the fact that PCR did pick up on rabies RNA on his body. So we're going to keep going to the next section. So we talked about ways to culture it, ways to identify viruses. Let's talk about other types of acellular infecting substances. So viroids, virusoids, and prions. So with viroids, these guys are going to be short strands of RNA, and they are able to self-replicate viroids. So there's different ones out there. There is the potato spindle tube viroid that clearly attacks potatoes, like in this picture. But there are also other ones that can attack avocados, peaches, tomatoes. These are also susceptible to a viroid, this little bitty type of RNA that is capable of copying itself in the organism. That's a viroid. Virusoids are going to be RNAs as well, but they're not self-replicating. What they have to do, they are going to require a helper virus. So if you got a virus and then you also have this virusoid on top of it, then you have more symptoms than you would with just the virus alone. So they have to have the helper virus. A virusoid must have a helper virus. There's only a few of them out there. We would lump them into the group called satellite RNAs. For us, the one that they mentioned in the textbook is hepatitis delta virus, and it goes along with the helper virus. Its helper virus is Hep B. So if you have Hep B and you also have hepatitis delta virus, which is a virusoid, your symptoms are worse than if you just had Hep B alone. So these things are single-strand RNA, but they can't self-replicate without that helper virus. And then the last one we're going to talk about are prions. And prions are not RNA, they are proteins. Prions are proteins. And it was first discovered in the 80s by Stanley Prusiner. And so he was studying scrapie, which infect sheep. And so he made this study, and back in the day, in the 80s, we said that there was no disease that didn't have a genome. It had to have some kind of genome. It's got to have RNA. It's got to have DNA, something. And Stanley was like, hold up, scrapie is not showing me any of those things. All I'm getting are these protein particles, but that's all I'm finding in this disease. It's got to be a protein. And he got some pushback for that. But eventually they came to realize that he's right, that it is just proteins. And so he won the Nobel Peace Prize in 97, so 15 years before his discovery was actually accepted, and he got the Nobel Peace Prize. But prions are proteins. They're proteins run amok. And what they do is they change your cells, they change the proteins, and they cause misfolding on them, and it causes plaques, which eventually will cause neural degeneration. on the brain. So it's called transmissible spongiform encephaly, TSE is what we call them. In people, there are a few of them: Kuru, fatal familiar insomnia. I'm going to slaughter this name here: Gershman Strausser Schrinker disease, and Creutzfeldt-Jakob, CJD, which is what we talked about in chapter 1, when Cora came in and she was showing these signs. She had CJD. And so what it is it gets transmitted sometimes in animals. We mentioned mad cow, scrapie for the sheeps, or chronic wasting disease that you'll see in ungulates, like deer. They can get chronic wasting disease. How do you get it? How do you get this disease that is fatal? How do you get these proteins? So you can get it from animal to animal by eating contaminated meats, or those animals could be fed bad feed that was contaminated with these prions beforehand, and this cow got cut up in the slaughterhouse, and those prion particles made it into the animal feed, and then every animal that ate that animal feed now gets that prion. Another way that you can get these prions are heredity. They can be passed down, or you could get it from contaminated tissue, like a blood transfusion or an organ transplant. These are fatal. There's no cure against prions. They will cause death eventually. Sometimes it's kind of rapid. This is what it looks like for a healthy brain, and this is CJD brain, and we can see it is shrunk in on itself. The plaques have started, the brain is shrinking up, and it's not a normal brain. It almost looks like Alzheimer's, with all the plaque formation. But this is, since it's not Alzheimer's and it's a whole bunch of proteins, this is CJD. This is mad cow. This is spongiform encephaly. So all of the different things in the table here, as far as humans and how you can get it. One of these includes cannibalism, so I don't think anybody will have to worry about that. But that's what Kuru is and how you can get it. All right, so let's follow up with David. What's going on with him? He's got results from the skin test, and the skin is negative. Skin test is negative, yes. The saliva sample, we said that they tested, was negative. But since his PCR, his RT-PCR came back, that they did find viral particles, they're going to continue to give him post-rabies exposure. So he gets the lovely experience of post-rabies vaccination. So it says he's going to get 4 shots in 14 days that will bind with the rabies and help eliminate it out of his body so it does not show signs. And it says that David makes a full recovery because he caught it in time. If somebody waits until it's too late and you're showing symptoms, it is too late. So this is a good story that David caught it in time and he was able to take treatment and get post-rabies exposure vaccine. So that is it for chapter 6. This is the last of the content that will be on the first exam test for you. So let me know if you got any questions and I'll get back with you.